Hey all, welcome to the 10th live session of Animal Physiology course taken by Professor Manak Das. So I'm also one of the TA for this. I'm Harsha. I'm a PhD scholar in Department of Biological Science in ISER Balambur. So, hi Amrita. I was just giving my self introduction only. Where are you from? Okay, in which course you are doing, where you are studying? Okay, yeah, so I will again repeat myself intro for you. I'm also one of the TA. I hope did you like have you attended my live sessions earlier? Okay, okay. So I am also one of the TA. My name is Harsha. I am a PhD scholar in Department of Biological Science in ISER Berambur. I am basically from Kerala. Okay. Yeah. So have you attended this week lecture? Oh, okay, okay. It's fine. You can whenever you get time, you can go through everything. So, if you remember, in the class, sir started with introduction to animal physiology, followed by then there was a lecture of series about different organ system, starting with the integumentary system, then going ahead to skeletal system. Skeletal system, it was almost explained about how skeletal muscles works in coordination with um, neuronal system and about how the muscles work, how the excitation, contraction, coupling, a short class there. Sliding filament theory, he explained very well. Then we went ahead to neuronal system where we again saw what are the structural details of neurons and what are the other types of cells present, how these what are the excitable cell types, how these neurons are able to perform in coordination, how action potentials are generated, what kind of receptors are there, then how through this receptor they are able to perform all this function. Then coming to multiple types of neuronal disorders, neurodegenerative disorders, Alzheimer's, Parkinson, amyotrophic lateral sclerosis. All this we have seen a brief introduction. Then finally, after discussing all these things, we went into discussing with different types of senses, that's such as olfaction, hearing, then about vision. Last week it was mainly about how the vision, how eyes are able to perform the function, all these things. Okay, so week 10 was mainly about the next. So we have done with the neural system. It was very long organ system classes. Now we have almost done with that. We went ahead to the cardiovascular system. So Sir was mainly talking about the structural details of heart, how heart is able to do the function and everything. Since you have not been went through the classes, don't worry. I will give a recap of each classes in single slide. Then we'll try to solve some question. If you don't know, also you can just type whatever the question you are guessing which is right. You can type that. Then we'll be discussing about it. Yeah. So I will present my screen. Okay. Yeah. So welcome to 10th week live session. Uh, Yeah. So cardiovascular system also there was five classes. First and second class was mainly regarding structure and function of the heart, especially regarding how many chambers we have, why specifically that chambers, how coordinatedly they are working, how the blood flow is regulated, why heart are able to pump in a auto-regulated manner, Then in the third class, mainly about conduction circuit. And coming to fourth class, he taught us about contractile system and conducting system in coordination. Finally, in the lecture 5, it was mainly regarding electrocardiogram and comparison of action potential between. There are two types of cells that are present in conduction system versus contractile system. So how the pacemaker cells or cardiomyocytes are different in terms of action potential. Yeah, so this structure and function of heart. In the first lecture, he taught us mainly about there are four chambers in our heart, starting with the right atrium. So whenever there are two vena cavas, or there are superior vena cava and inferior vena cava. So these vena cavas collect all the impure blood from different parts of the body. Superior mainly collect from the upper part of the body, and inferior mainly collect from the lower part of the body. Then this blood will goes and comes and fills in the right atrium. So right atrium to right ventricle. In order to prevent the backflow, there is a valve. So there was a interesting questions are asked. In one part it is there are something triple valve, and in one place there are two valves. So it was an activity question he gave us to find out where is tricuspid and bicuspid. And I think in the third lecture he himself told that where is the tricuspid and bicuspid. So remember, from right atrium to right ventricle, in order to prevent the backflow of the flow of the blood, there is tricuspid valve is present. So then from right atrium to right ventricle, so blood flows. Then impure blood will go to pulmonary artery. So this is the only one artery that carries impure blood. Pulmonary artery will take the blood from the heart and goes to lungs. There it will exchange CO2 and refills new oxygen. Like that, it will take the new oxygenated blood and gives to again to left atrium. So left atrium to left ventricle, there is again one more valve, bicuspid valve, which to prevent the backflow of the blood. So now oxygenated blood comes from the lungs to left atrium, then to left ventricle. Even though I said this order, actually the series, if you are looking time wise, the uh, events are not happening like this. So there are something that is happening like at the same time almost. So basically, the blood comes from all the vena cavas to right to atrium, and blood comes through pulmonary vein from the lungs to left atrium. Is around there is little bit time only. So first blood fills in right atrium, then blood fills in left atrium, then right ventricle, then left ventricle. That start explain more in the third week of third lecture also. Okay, so this kind of circulation is also known as closed loop system. Why? Because it's not like cockroach body, right? If you are opening, I in plus one plus two we have to do the cockroach dissection. I don't know you guys have done it. So if you dissect a cockroach and see, we can see everything inside cockroach body is dipped in some white fluid. That is basically open heart. But if you're looking at in ourselves, everything is very safely secured and chambered, and everything is in a very organized manner. So that kind is known as closed loop system of blood flow. Our doesn't flow in a open system. There are blood vessels, closed blood vessels. Inside that only blood is flowing. So it's like known as closed loop system of blood flow. Yeah, so that about the first lecture. In the second lecture, it was a continuation of the first lecture. So, which I told just now, the time event, even though it's happening in that order, it's different. So, first what, always remember event means blood getting filled in right atrium. E2 is blood getting filled in left atrium. E3 is blood getting filled in right ventricle, and E4 is the blood getting filled in left ventricle. Okay. Then so whatever we are saying about the pumping, it's about the mechanical contraction that is happening. So there is a node present in that which regulate, okay, the auto regulated pumping of the heart. So starting with that is SA node. So that is basically present in the right atrium. then the blood flows through like this, and then from the right atrium to the center part like this. So again, this is right atrium. So this is E1 and this is E2. So if you compare from the blood to reach here, it is a shorter pathway than the blood to go through this way to coming here. So that's why first E1 to E2, then to E3, then to E4. So remember this thing also. So and also remember sinoatrial node is the one who regulates the pacemaker activity of the cell. Pacemaker activity means the auto-regulating thing happening in regulated mainly by sinoatrial node. Yeah. So anything else? And please remember that E1, E2, E3, E4, which event is coming happening at what time? E1 right atrium first, then left atrium, then right ventricle, then left ventricle. So E1, E2, E3, E4 also, please keep it in mind. Um, and interestingly, in the third week, when we talk about the mechanism, sir also told us, uh, we have seen in the last week when we talked about our photoreceptor cell, we have seen they are also situated at a resting membrane potential of minus 40 millivolt. Like that, if you ask a question, since our most of the cells are situated at resting membrane potential of minus 70 millivolt, but then how heart is able to keep on pumping? So there must be some additional mechanism present in them. That's why they are able to do that very without any rest, if we basically say. That's right. If any injury or accident happens, sometimes brain death also happens. Then we consider that brain death. So he lost ability to everything. Still, heart is beating means that person is alive. So when we consider a person is completely dead if only heartbeat stops, right? So that heartbeat is that much important. Yeah. Uh, so remember that its action potential generation, the way they work is little bit different. Remember they are situated at a resting membrane potential of minus 40 millivolt. Okay. Now in the week third lecture of week, yeah, this one. Third week, it was mainly about conduction circuit. Uh, Sir made us, like Sir was telling about what are the different types of cell types that is present. So now you remember conduction system is the pacemaker cells, mainly coordinated by pacemaker cell. Then around the pacemaker cell, there are other types of cells that is present in heart, which is called as cardiomyocytes, which is a part of contraction system. Okay, conduction system represented by pacemaker cells, contraction system represented by cardiomyocytes. Other than that, definitely bloods are coming, bloods are flowing. So in order for that, there are blood specific endothelial cells, blood vessels, everything is there. Then there are some stem cells are also present in heart. So remember all these things. Yeah. So, since the conduction activity of the heart is happening in coordination with contraction activity, that is why it is electromechanical system. So, electromechanical system. Please remember this, okay? Electrical activity is happening in coordination with mechanical system. So, pacemaker cells, what they do, there are spontaneous action potential generation, which leads to activation of surrounding cardiomyocytes around the pacemaker cell, which again leads to generation of action potential in cardiomyocytes. So that's why electrical activity leading to the mechanical activity. So that's why it's known as electromechanical system. Uh, Now there is a concept of calcium. Why? Because Sir told us since it is situated at minus 40 millivolt. Let me open the pointer. If you have any doubt or whatever I am telling not understanding, please stop me and feel free to ask. Okay. So you can see Now you are understanding, right? Whatever I am telling. Yeah. So the action potential is at minus 40 millivolt. Usually it is at minus 70 millivolt. So usually what happens in order to there is a threshold frequency comes whenever it is at minus 30 or minus 40, then the gates are open, ligands are open. So the inflow of sodium comes and that's why it gets into positive. But here it is already at minus 40 millivolt. So what happens? If it have to stay in a particular point where it has to stay above the positive, like above the positive axis and for some time, then sodium is not only required because sodium can make it activate only from the minus 30 to positive. It's already at minus 40 and it has to shoot above again above positive. Then there must be the presence or activity of some more ions. So that ion is calcium, which is released from the sarcoplasmic reticulum. So, sir explained more about this in the next lecture also. Uh, yeah. So, and in the fourth and fifth week, it was mainly regarding through slide presentation. He again taught us whatever he said, taught us in the last 2 weeks. So remember this. I will tell how the exact activity is happening. So there is activity of sodium ions usually happens, but here after that, when it reaches at some particular plus point, calcium will be opening and calcium ions will open and calcium ions will this line you see, the green steady line over the positive axis, that is happening because the activity of calcium. So calcium ions opens and there is a some techniques in sarcoplasmic reticulum. It opens and it closes. So when it closes, again our old normal activity that happens in action potential steps will happen. Potassium ion channels will be then depolarization, hyperpolarization, everything will occur. Only one difference is there is a long persistent positive axis or steady state you can see that is due to the inflow of calcium from sarcoplasmic reticulum. Okay, so then this is actually how it looks like even though we when we draw we look draw like that. So there are four chambers: right atrium, right ventricle, left atrium, left ventricle. So these are just screenshot I took from SARS class. You can go back and refer to the lectures and the original lectures, so you can then you can learn it from there. So this type of is also we already told these are also known as closed circuit system. So yeah, this is the way it passes: right atrium to right ventricle, then left atrium to left ventricle in the order wise, not in the time wise. Time wise, always remember it is even means the blood filling in right atrium E2, left atrium E3, right ventricle E4, left ventricle. No change in that. Okay. Yeah, then SAR gave a link in the NIH. There is a NCBI like site. You can see how this is working. So I will open one by one, then we can see. Yeah, so do you understand the pathway, how the blood transfers from the video? Yeah. Now there is one more video about valves of the heart. Let's see that also. Then we'll go to the next session. So that is about four valves: tricuspid, bicuspid, or mitral valve. Tricuspid is between right atrium and right ventricle. Mitral valve is between left atrium and left ventricle. Then there is one pulmonary valve between pulmonary artery and right ventricle. Then there is one aortic valve. So wherever, in order to prevent the backflow, there are valves. That's it. And remember what valves in which area. Okay. Now, in the fourth and no, in the fifth week, it was mainly regarding electrocardiograph, EKG. What are the uses of EKG? So he has always most of the slides were mostly useful. So please check into the notes and study everything. Okay. They again went through what are the uses of EKG. It tells us how the normal activity of the heart going on. If there is any variation or there is any pressure block or blood flow is not happening regularly, then there will be disrupt in these kind of graphs. That's why in heart attacks, like people comes and any heart issues, doctor feels doubt, they will go tells you to check for a EKG. Okay, then remember P wave represent atria, QRS ventricles activity, T wave usually represent it at rest. P, QRS, then T. Okay. So in one of the question it is coming which is what activity. So in that time I have given the screenshot of Sar's slide where he is telling what wave is corresponding to the which activity of atria. So again about the action potentials in cardiomyocyte, remember the difference between pacemaker. Pacemakers are like very short waves. It's like always going because pacemaker is always regulating, right? So it cannot slowly comes up then stays like this. No, it cannot do that. So it's always activated. But in cardiomyocytes, the waves will be like the slide I showed yesterday. 1 minute. No, slide I showed just before this one. This one. So this is how cardiomyocyte represent action potential. This is how pacemaker cell. Remember always pacemaker should be activity should be continuous. There should not be any lag. That's why they are auto-regulating. But in cardiomyocytes, it should be like this. So due to the activity of calcium. Yeah. And that calcium is released from sarcoplasmic reticulum. So now let's go and move and try to solve some questions. So I will open the in-call messages so that I can see what you are typing in the chat box. Question number one. In which of the following is not a component of blood? Option A, white blood cell. Option B, platelets. Option C, red blood cell. Option D, astrocytes. Not a component of blood. Good. So we know, you know what are the functions of all the other three, right? Yeah. Simple question. So blood is a specialized body fluid and it has mainly four components. Plasma and the associated cell, mainly two components if we count all the cells as cells. So plasma is the uh, portion that is devoid of cells. When you do some research or experiment, mostly anything related like that, we always prefer to take the blood plasma. Not in the experiment which you want to detect red blood cells or white blood cells activity, but if you want to store it for a long time and want to check the gene expression analysis or something like that, then we go ahead with the plasma. So red blood cells, we know they are the contain hemoglobin and they has the hemoglobin adapted in such a way that they can bind with oxygen. White blood cells are again, there are five subtypes, granulocytes and agranulocyte. Each cells are specified for specific function. They deal with the immunity, T cells, leukocytes are there. Lymphocytes, leukocyte is WBC is one another name. Lymphocytes are there, which deals with immunity. T lymphocyte, so there are a lot of innate immunity, adaptive immunity. So specified for T lymphocytes and B lymphocytes are there dealing with each part. Then there are monocyte, basophil, eosinophil in response to allergic response. Um, so like these specific functions are there. Platelets are mainly for the blood clotting, right? They will otherwise what happen if platelet is not there? There is no one to go and trap our blood in such a way. Then blood keeps on flowing and we will die. So blood clotting is a very effective mechanism that help us to conserve our blood in any kind of injury. Uh, so astrocytes, what is astrocyte? Where we can see astrocyte? Okay. You don't remember when we talked about the neuronal system and everything. Do you remember any astro sites that we learned there? Okay, go back and refer. Okay. There other than neuron, there were some glial cells, and if you remember, what are the subtypes there? I'm not telling. Plasma is a component of, again, very simple question. Just now discussed. Option A, blood. Option B, lymph. Option C, both blood and lymph. Option D, none of the above. Yeah. Can you please type it in the chat box also? Because I will be uploading this in the YouTube. So, uh, yeah. Yeah. So, correct. Plasma, we just now heard there are two components, plasma and cells. So, plasma is a component of blood. Vena cava is a dash that carries dash. Option A, artery, oxygenated blood to heart to the body. Option B, vena cava is a artery that carries deoxygenated blood from the heart to the body. Option C, it's a vein. It carries oxygenated blood to the body from the heart. Option D, it's a vein. Carries deoxygenated blood from the body to the heart. What is the correct answer? Vena cava, superior, inferior is there. What is the function? What is its subtype and what is it carrying from where to where? So which option? Okay. Yes, correct. It's a vein. There are two types, superior vena cava and inferior vena cava. And superior vena cava collect impure blood from upper part of the body. Inferior vena cava collected from lower part of the body. Remember that video we saw also? So that will supply it to the which atrium? Which chamber in the heart? to the right atrium it gives. Okay. So you don't have to type the entire answer. You just have to choose the option and say it's A, B, C, D, or D in the chat box. Okay. Next, aorta is the main dash originating from dash. Option A, artery originating from left ventricle. Option B, is it a artery originating from right ventricle? Option C, is it a vein originating from right ventricle? Option D, it's again a vein originating from left ventricle. So the name itself indicate, right? Aorta means artery and it's originate from the left ventricle. Carries pure blood or impure blood or oxygenated blood or carbon dioxide mixed blood. What is the correct answer? Aorta carries always oxygenated blood, right? Oxygenated blood from the left ventricle and supplies it to the rest of the body. Uh, so lower left chamber of your heart. Next question. Purkinje fibers have dash and are dash than cardiomyocyte or myocardial cell. It's about comparison of how much myofibrils are they have more or less compared to cardiomyocyte. Also their size. Are they larger or smaller? So remember in the ventricle part there are a lot of Purkinje fibers and how they are comparatively compared to cardiomyocytes. So Amrita is saying they okay. Let's see. Huh. How do you know this? Okay, very good. So remember, Purkinje are always larger than cardiomyocytes and they have fewer myofibrils. Okay. Um, so how they are distinguishable histologically? Because they lack transverse tubules. They lack transverse tubules, but that are present in myocardial cells. If you are comparing, Purkinje are larger, they have fewer myofibrils and have no transverse tubules. Because of their primary role in rapid conduction of the electrical impulse, Purkinje cells are fewer. That's why they have fewer myofibrils because they have to like electrical impulse has to be transmitted very rapidly across them. So they have very fewer myofibrils than myocardial cells. Now the question is just the extended part of the previous question. Which of the following do not have T tubules or transverse tubules? Option A cardiomyocytes, option B Purkinje fibers, option C cardiomyocytes and Purkinje fibers both do not have, option D none of the above. Hmm? No, no, I'm not surprising. Just now we discussed it actually when I explained about Purkinje fibers. It's fine, it's fine. You can choose any option. Uh, that is correct answer only. Purkinje fibers do not have a T2 port. Okay, so explanation I said here. Here, Purkinje cells lack transverse tubule. Transverse tubules are only present in myocardial cells. Okay. Now it's about the conduction system, not contraction, conduction system of the heart. Which of the following is not a part of the heart's conduction system? Not a part. Okay, not a part. Remember, not a part. Option A, AS junction. Option B, AV node. Option C, SA node or sinoatrial node. Option D, bundle of His. Okay, let's see. Yes. So, what are the cells that are part of conduction system? SA node means sinoatrial node. Origin is that. Then it will transfer to atrioventricular node or AV node, atrioventricular node, followed by bundle of His or atrioventricular bundle, and then Purkinje fibers. So these are the four components: sinoatrial node or SA node, AV node, bundle of His, and Purkinje fibers. AS junction is not a part. Which of the following in the heart's conduction system is the primary pacemaker and generates the initial electrical signal? Option A, AS junction. Option B, AV node. Option C, SA node. Option D, bundle of His. Initial point of the conduction system. What is the origin point? This I showed when we talked about that brief introductory lecture time also. There is the one thing present in the right atrium. What is that node? Uh, Correct answer is SA node. Okay. Sinoatrial node or SAN or SA node is the primary pacemaker of the heart. Here. This is the screenshot from SAR's slides that he uploaded. Okay. Origin is sinoatrial node going to atrial syncytium, junctional fibers, atrioventricular node or AV node, going to AV bundle, going to bundle branches, going to Purkinje fibers, finally reaching ventricular syncytium. In electrocardiogram, QT wave signifies what? Option A, depolarization of the atria. Option B, initiation of signals in left atrium. Option C, electrical activity in the ventricles. Option D, repolarization of the ventricle. Any idea? You can guess. Okay, let's see. So, QT interval represent the duration of ventricular electrical systole, which includes ventricular activation and recovery. So, yes, again, your guessing is correct. You guessed the electrical activity in ventricle only, right? So your guessing even is getting correct. Yeah, so this is again a part of the screenshot I took from the lecture. So P wave indicate depolarization of the atria. This please remember all these phases, what is its function and time duration, because such type of questions have seen in the assignment also. So P wave represent depolarization of the atria. PR interval represents time from the onset of arterial depolarization or P wave to onset of ventricular depolarization. So remember P wave at arterial depolarization and QRS ventricular depolarization or depolarization of the artery is P wave. QRS is depolarization of ventricle and PR interval is the time from P to QRS. So the time duration of arterioventricular conduction is around 120 to 200 millisecond. QRS complex uh, stays for less than 120 millisecond, which detects the activity of depolarization of ventricle. QT interval detects depolarization and repolarization of the ventricle. So QRS depolarized completely. Now it's coming back and then repolarization till it will complete the repolarization. So it's generally less than 420 millisecond in a man or potentially less than 440 millisecond, 40 millisecond in woman. RR interval is the duration of ventricular cardiac cycle and indication of ventricular rate. P wave, arterial depolarization or depolarization of the atria, QRS depolarization of ventricle, then all this interval. PR means time from P to QRS. QRS is generally less than 120. QT means depolarization and repolarization of the ventricle that together will be less than 420 millisecond average. RR is the duration of ventricular cardiac cycle. Why did I tell this again and again? Because there are questions coming with this. Now there is a tricky question. A medication X generate the QT wave of 200 millisecond. A medication X generate QT wave of 200 millisecond. So option A, medication causes QT wave prolongation. Option B, medication causes shorter QT wave. Option C, medication causes normal range QT wave. Option D, this is not enough information to make a conclusion of size of QT wave. So for this question, you should know what is the general time taken for a QT wave. Then we can answer. Okay, option A. Is it a guess or you note down the time point? Okay. So, but correct answer is B. Did you said B only? No, this time this is not correct. Medication causes shorter QT wave. Why? If you are checking the average time that we discussed, this one. So, what is the QT interval time? 420 millisecond, right? So here the question, it generate only 200 millisecond, which means it is very lesser than the normal QT wave. So that's why it said medication causes shorter QT wave. So just the question's idea is we should know what time is it will take for each cycle. So correct answer is B. So next question. Dash begins slightly before ventricular systole. Dash begins slightly before ventricular systole. Option A P wave, option B QRS wave, option C R wave, option D T wave. Okay, let's see. So when we discuss that black slide, remember ventricular systole determines the depolarization of the ventricles, right? So ventricular systole means QRS wave only determine that, right? So the end point of the P wave depolarization is the start point of the arterial stage of the systole. So P wave is the activity of depolarization of arteria. So the P wave is ending means that will be beginning with the starting point of arterial stage of systole. Ventricular stage of systole begins with at the R peak. You are right. It the peak is at R. Like peak will be at R, but question was dash begins slightly before the ventricular systole. So in the R it was already peaked. So according to that correct option, you asked QRS wave. Okay. And T wave means end of ventricular contraction, after which the ventricular relaxation comes. So remember, P wave means atrial depolarization, QRS means ventricular depolarization. P wave ended with articular uh, depolarization. So endpoint indicate arterial systole. Then QRS wave. R is the peak point of the ventricular systole. T wave means ventricular contraction. End of the contraction. Again, after T wave, that is ending with the ventricular relaxation. So should I go back and again check what is PQ? You remember the each wave, right? Should I show a image? If you want to see, I can go back and okay. Ah, here there is. Remember what is P? This one is P. Okay, means arterial systole, depolarization of the atria. QRS. QRS indicate depolarization of the ventricle. This one is the T wave. So connecting to this, we are saying T wave is the rest. By that time, ventricular relaxation will be occurring. This is will be ventricular systole is happening. But the peak is R. P wave, arterial systole. So I'll go back to question. Understood now? P, Q, R, S, T. 1 minute. Actually, someone opened the door, like from outside. Door. Someone opened. It's like open. I don't know who was there. Oh, next question. A dash is an instrument that records by the dash the duration of the interval between the successive QRS complexes in the electrocardiogram. Option A, instrument is EKG that records by the height of the successive spikes. Option B, instrument again EKG that records by the width of the successive spikes. Option C, it's cardio tachometer, which records by the height of the successive spikes. Option D, it's an instrument of cardio tachometer again. That will be records by the width of the successive spikes. So what is the correct option? Remember duration. Okay. Uh, so what is EKG? EKG determines all the P, QRS and T. Here the question is that only determine duration of the interval between successive QRS complexes in electrocardiogram. Let's see, I don't know. So correct answer is cardiotactometer only. Electrocardiogram name you remember P is there, then QRS is there, then T is there. Here, specifically it is telling it records duration between two QRS cycle. Okay, so and it records by the height of successive spikes. So correct answer is option C. Cardio tachometer. If you want to see how the instrument look like, I don't have an image, so I'll take from the internet. This one. Can see, right? This is cardio tachometer. Next question. Seeing the question, don't worry. It's just read carefully. It's not only from the yeah. I will read. Read the following paragraph carefully, then read the statement. So, the widely recognized importance of scale in determining function in mammals has led researchers to investigate physiological processes at the extremes of body mass. From the smallest shrews to the largest whales, physiological performance at the extremes may be shed light on constraints to body size. In particular, understanding cardiac function at these extremes remain a central challenge in physiology, especially as it relates to energy demand in the natural environment. At the larger scale, marine mammals exhibit a suite of anatomical and physiological adaptations to enhance diving ability and improve the foraging capacity during a breath hold. So each and every mammals have its own adaptation from the scale to each part to enhance its diving ability or for example foraging capacity so that the survival of the fittest will be keep on happening. Now there are the statements are option A and not option A. First statement is decrease in heart rate associated with cardiovascular dive response in marine mammals result in heart rate during dives that are lower than resting values, whereas the sea surface the heart rates are higher. That was about cardiovascular dive response of marine mammals. They are saying it decreases in the diving time and at the surface time, it is heart rates are usually higher. Second statement is a slower heart rate during dives decreases oxygen delivery to the tissue and slows the overall depletion of blood oxygen stores while also decreasing the oxygen consumed by the heart itself. So second is the explanation kind of first statement. Why they have slower heart rate during dive? It is to decrease the oxygen delivery to tissue and thereby deplete the blood oxygen stores and also for decreasing the oxygen consumed by heart itself. C is the hypothesis is that exercise modulates the dive response and increases heart rate during the dive is particularly relevant to extreme nature of blue whales because feeding lunges have an estimated metabolic rate 50 times the basal value. So they are saying this is again a response that is taken by the blue whale for feeding. Why? Because feeding lunges have a excess metabolic rate of 50 times. So in order in that time they should do this, then only their foraging capacity can be in this way they can increase it. D, the lunges are considered to accelerate the depletion of body oxygen stores and account for shorter than expected dive duration. Elevated heart rates and increased blood to muscle oxygen transfer during a lunge may be critical because of the metabolic cost of a lunge and low myoglobin concentration of blue whales relative to myoglobin concentration around 5 to 10 times higher in most marine mammals. Let me drink some water. Statement is exercise depth and volitional or anticipatory control have been postulated to modify heart rate during the dive response via the autonomic nervous system. Increased heart rate with exercise may occur via withdrawal of parasympathetic run or increased sympathetic run. Plunge compression during descent and expression during ascent may also contribute to decrease or increase respectively in heart rate via pulmonary stretch receptor reflex. So in most of the cases, like this much bigger question comes, that is like giving a more information to you, because we have not discussed this in class, so that by giving this kind of question, they are giving lot of information to you. So the question is to just find out what are the correct statements. So first option is B only true, B is not true and all the rest are true. Second option is A is not true but all the rest are true. C is all are true. D is except E all are true. What is the correct? So all the statements are actually true. This is taken from a article, so you can refer if you want to look into this article. Okay. A, B, C, D, E are true. So in such kind of thing, it's just a information providing. But read like at least know these are the things there. So all the statements are true. Next question. Bradycardia is a dash than normal heart rate. Option A, bradycardia. Higher. Option B, slower. Option C, first slower followed by higher. Option D, extremely higher. What is the correct? Bradycardia. So correct answer is slower. Okay. Is a condition where your heart beats more slowly than expected under 60 beats per minute. Sodium potassium pump exchanges dash amount of potassium in replacement of sodium. Option A 2 2, option B 2 1, option C 3 2, option D 2 3. What is the correct answer? So correct answer is, remember, 2 potassium in replacement of 3 sodium, right? 2 potassium inside. So first depolarization, then due to the sodium pump, again it will try to go outside, that it will make repolarization happen, it will make more negative, which is hyperpolarization. Then to make it again back to normal minus 70 millivolt, sodium potassium pump will be opening, so it will push us out 3 sodium out and takes in 2 potassium inside. So 3 potassium out and 3 sodium out and 2 potassium in. So correct answer is 2 and 3. D. Should I draw? Then I can draw. Suppose these are the sodium potassium pump. There are membranes. Okay. Remember their choline attached tails are there. Both sides are it is there. I will take an eternity to draw both. So I'm not drawing correctly. Remember it's like this, then this side also. Yeah. So phospholipid bilayer, this is a sodium pump. So now this is inside, okay, this is outside. Outside, inside of cell. So usually who is outside? Sodium. So now sodium is more inside. So they want to push out sodium. So 3 Na plus will go. 3 Na plus will go out. Yeah. 2 sodium will come inside. 2 A plus. Yeah. So that's why 2 potassium in exchange of 3 sodium. Option D is the correct answer. While measuring blood pressure, 120 mmHg is dash and 80 mmHg is dash. So this consider as ABCD, okay? A is systolic pressure is 120 mmHg and 80 mmHg represents systolic pressure, both systolic pressure. B is systolic pressure and diastolic pressure. C is diastolic pressure and systolic pressure. Option D, both are diastolic pressure. So what is the correct option? Correct answer is 120 mmHg is systolic pressure and 80 mmHg is diastole. Systole represent the contraction, right? So contraction time, it definitely require more pressure. So that's why 120 by, whenever we want to check blood pressure, 120 by 80 you can see. So 120 means systolic and 80 represent diastolic. If heart rate is 72 beats per minute, duration of cardiac cycle is option A 0.833 second per beat, option B 0.0833 second per beat, option C 0.0139 second per beat, option D 1.39 second per beat. It is simple mathematics. So 1 minute means 60 seconds. So what will be the seconds per beat? Correct answer is 0.833. How did I understood that? So they are saying there are 72 heartbeats per 72 heartbeat per minute. So 1 minute means 60 seconds. So in 60 seconds there is 72 beats. So just for duration of one cardiac cycle, we just have to divide 60 by 72, right? 60 by 72 is 0.833. So it's simple. Remember 1 minute 72 beats per minute, which means convert minute into second, that is 60 second. So one duration is equal to 60 by 72. Let me use the pen. Yeah. So is equal to 60 divided by 72. This will be 0.833. So duration of one cardiac cycle is per beat they have said, right, in option. That's why we did 0.833. You calculate if you have doubt. So correct answer is option A. Then if you are dividing it into each even, then remember duration of total cardiac cycle is 0.8. In that, arterial systole is 0.1 second. Arterial diastole is 0.7 second. Ventricular systole is 0.3 second. Ventricular diastole is 0.5. That's why adding both together it will be 0.8 only, right? A single cycle of cardiac activity can be divided into two basic phases, systole and diastole. So arterial systole and ventricular systole are there. Arterial diastole and ventricular diastole are there. Remember the activity first happen right atrium systole, then at the same time um, left atrium blood fills in, then again left atrium, then again left ventricle. The difference between action potential generated in cardiac and skeletal muscle is that the onset of action potential, the permeability of dash decreases fivefold in. So this is between cardiac versus skeletal muscle. So you have to compare the permeability of which ion decreases fivefold in which cardiac or skeletal compared to the other. Option A, sodium. Option Sodium and it decreases fivefold in cardiac muscle membrane. Option B, decreases the permeability of potassium and it decreases the fivefold in skeletal muscle. Option C, it's due to potassium, decreases fivefold in cardiac muscle. Option D, sodium, it decreases fivefold in skeletal muscle membrane. What is the correct answer? Correct answer is option C. Okay. The permeability is due to potassium ion and that permeability decreases fivefold in cardiac muscle membrane compared to skeletal muscle. Why? Because during phase 0, membrane permeability to potassium decreases and fast sodium channels open. That we know because potassium has to decrease and sodium open. That's why positive charges will come inside and threshold it will reach. Then it will start shooting the action potential, producing rapid depolarization from, that's why depolarization happens from minus 90 to 10 millivolt. Now if we compare during phase 1 there is a partial repolarization because of a decrease in sodium permeability. Because now sodium has rushed inside and it's closed, sodium ions has closed coming inside, so there will be a partial repolarization. Phase 2 is the Plato phase. You remember this chart? Yeah, it looks like this. Again, again it will go. So this is the in cardiomyocytes. So this straight phase they are talking about. This straight phase is due to the Plato phase, and Plato phase is due to the rushing of calcium ion from sarcoplasmic reticulum. and sarcoplasmic reticulum takes back the calcium ion. That time it will again goes down. So phase 0, remember depolarization. Phase 1, partial repolarization due to sodium closed. Now calcium comes inside, so there is a steady phase. And prolonging the action potential, membrane permeability calcium decreases somewhat towards the end of phase 2, and the plateau is partially maintained by an inward sodium current. Sodium flows into the cell through sodium calcium exchanger. The exchanger transfers 3 sodium ions into the cell in exchange of 1 calcium ion flowing out, and so produces a net inward positive current. Now this steady phase towards the end, it is also due to the sodium, because sodium also starts coming inside, and the exchange pattern is like um, here. Sodium flows into the cell through sodium calcium exchanger. So that exchanger transfers 3 sodium into the cell exchange for 1 calcium. Now calcium will go out and sodium will come inside. 3 1. And so produces a net inward positive current as calcium channels inactivate towards the end of this plateau phase. So towards the end, calcium has closed, and inward potassium current produces repolarization. So this is happening due to the normal repolarization, which is due to the potassium current, right? Inward potassium current, potassium starts coming, and the resting membrane potential in phase 4 is approximately minus 90 millivolt. This is produced mainly by selective permeability of the cell membrane to potassium and concentration gradient for potassium that exists across the cell membrane and is close to Nernst equilibrium potential for potassium. So remember, zero depolarization of sodium, uh, 1 a sodium comes in, that's why depolarization happening. Now in phase 1, repolarization due to sodium closed out. Now calcium comes in, so steady phase will keep on coming. Calcium influx, positive charge will coming in. Along with that, calcium will start going out in sodium calcium exchanger. So 3 sodium comes in, so the steady phase is maintained. Here again, repolarization happens due to the activity of potassium current and repeated. So remember, correct answer is the difference between action potential generated in cardiac and skeletal muscle is that after the onset of action potential, permeability of potassium decreases 5-fold in skeletal muscle membrane. In cardiac muscle membrane. Potassium decreases in cardiac muscle membrane. Otherwise, it will happen like the normal cells, which maintains the membrane potential at minus 70 millivolt. So here, due to there is calcium's interplay and permeability of potassium is decreasing in cardiac muscle membrane. That's why it's able to maintain this pattern. Next question. More explanation here. If you know about the phase, this is the 0, 1, 2, 3, 4. Excess dash in extracellular fluid causes heart to become dilated and heart rate. So, which ion is higher in extracellular fluid, that will be causing the heart to become dilated. As a result, heart rate will be slows or fastens. So, option A, sodium or slows. Option B, potassium slows. Option C, potassium fastens. Option D, none of the above. What is the correct answer? Correct answer is potassium. If potassium is going out in extracellular fluid, and if there is a huge amount of potassium in extracellular fluid, that will cause heart to become dilated, and as a result, heart is dilating, right? So heart rate will be slowed down. B is the correct answer. Dash and dash regulate calcium movement in cardiac muscle. Option A, ryanodine and DHPR. Option B, TTX independent and TTX dependent channel. Option C, digitoxin and saxitoxin. Option D, aspirin and nicotine. This we have discussed in the skeletal muscle part. Towards the end, not in the skeletal muscle, in the neuronal end when we talked about contraction coupling. Which are the receptors that regulate calcium movement? That time we saw in response to skeletal muscle, flu is the same Receptors are activating in response to cardiac muscle also. There we learned about a calcium regulator receptors. What are that receptors? Correct answer is ryanodine and DHPR. Okay. So that we already discussed in those time questions, so you can refer back. Next question. Pacemaker cells maintain their resting membrane potential at dash. It is about pacemaker cells. Remember pacemaker cells is the question. 80 millivolt minus 40 millivolt minus plus 40 millivolt. Fourth is minus 80 millivolt. It's about pacemaker cell, not about cardiomyocyte. So what will be the correct answer? Minus 40 millivolt, right? Remember we discussed that's why they rapid. It has to put up positive, then again comes to normal, then again. So the threshold attains faster and the auto-regulatory manner is keeps on going like that. That's why it's auto-regulated. It keeps on pumping. So pacemaker cells resting membrane potential is at minus 40. Electrical activity of conduction circuit of heart starts at. Same question we discussed. I don't think any explanation needed. Electrical activity of conduction circuit how heart starts at dash. Option 1 SA node, option 2 AV node, option 3 aorta, option 4 vena cava, which is the correct answer. Correct answer is sinoarterial node. Okay. Calcium is regulated in cardiomyocytes in which organelle? Option A nucleus, option B vacuole, option 3 Golgi apparatus, option 4 sarcoplasmic reticulum. Where is calcium flow regulated? This we saw in the skeletal muscle also. Here there is some adaptation that it will takes back immediately. That's why the flow is maintained like the in a steady state is maintained. But the organelle is same. Calcium regulator. Correct answer is option 4. Sarcoplasmic reticulum. Carbon dioxide oxygen exchange in the body happens at dash. Option 1 nephrons, option 2 capillaries, option 3 hair, option 4 pacemaker cells. From the option itself, you can choose only one option. Its options are that simple, so you can choose correct answer. Carbon dioxide oxygen exchange. You can either attend the question by knowing the correct answer. If you don't know the correct answer, then you can try to rule out the wrong option so that you can find the correct answer. So let's do by the second method. Rule out the wrong option. Nephrons. Nephrons are basically parts of functional units of kidney, which my research interest is in. Third option is hair. You know, hair. Do you think inside hair carbon dioxide and oxygen exchange is happening? Four is pacemaker cells, which is the origin of the conduction, like the pacemaker, which is the heartbeat is happening continuously due to there only. So that is also not. So which is the correct? Capillaries. Okay, in capillaries, exchange of oxygen with carbon dioxide is happening. Which of the cells are found in the heart? Endothelial, cardiomyocyte, cardiac, pacemaker cells, all the above. I have given the figure also. What is the correct answer? Endothelial cells definitely because the blood flow is through there, so we can see endothelial cells doing blood like covering that part. Cardiomyocytes definitely because around the conduction system, cardiomyocytes are present. This is the arterial cardiomyocytes. Cardiac pacemaker cells which is represented here as conduction cells. So pacemaker cells are also there. Other than that, there are some fibroblasts you can see, some ventricular cardiomyocyte you can see, some smooth muscle cells you can see. All of these things you can see. All of the above is the correct answer. Which of these has highest diameter? Option 1 aorta, option 2 capillaries, option 3 pulmonary artery. This also when sir taught in the class, he specifically told when he mentioned about aorta, this is, I told the answer also accidentally. Yeah. So obviously correct answer is 1. Aorta has the highest diameter. Okay. Which of these has valves? Option 1 artery, option 2 veins, option 3 alveolus, option 4 branchioles. These two are part of lungs. So which of these has valves? Remember, wherever valves present in the heart, connecting to that also you can say. So veins have valves. Okay, this is why the figure is there. Vein only have valves. So correct answer is 2. Occurrence of two successive P wave means P waves is happening very one after the other. That means what is the problem? Option 1 left atrial enlargement. Option 2 left ventricular enlargement. Option 3 both A and B. Option 4 none of the A and B. So P wave, remember what is the activity of P wave? It detects atrial depolarization, atrial depolarization, which means one after the other means left atrial enlargement, right? Huh. So left atrial enlargement means it's the first P or mitral wave that will be left manifest as broad torched or bifid P waves, particularly in leg 2 of the ECG. Prolonged P wave duration. These P waves are prolonged, lasting longer than 110 to 120 milliseconds. That's why one after the other. Biphasic P wave. Left atrial abnormalities also often cause a biphasic P wave in lead 6 with an enlarged terminal negative portion. Left ventricular enlargement, it's not an option because that is typically indicated by QRS complex, such as increased amplitude of R wave or rather than P wave. Since P is definitely dealing with atria, from the option we can correctly say correct answer is left atrial enlargement is depicted by two successive P wave. P wave with decreased amplitude can indicate hypokalemia, hyperkalemia, hypocalcemia, hypocalcemia. So P waves duration has decreased, which means what is decreasing? Potassium or calcium, or is it increasing? What is it increasing? Is the correct answer. What is the correct answer? Correct answer is hyperkalemia, which means if P wave shorts in amplitude, that means potassium is higher. Okay. If potassium is decreases, what will happen? P wave will be causing abnormalities, and also P wave will be showing a higher peak. In the question it was decreased. That means potassium is coming higher. If potassium is lower, it will show opposite. It will show a higher amplitude or P wave abnormalities can be also seen. If calcium is decreasing, then it will primarily affect QT interval. So that will lead to more QT that doesn't directly do anything with P wave. If calcium is higher, then similar to hypocalcemia, it is again associated with QT wave only. So it does not directly alter the P wave. Yeah, so this in hyperkalemia, you can see a lesser P. In hypokalemia, you can see a more P. And in hyperkalemia, P is getting thoda sa bigger. In hypo, it is decreasing. So opposite, opposite. Again, it's about ryanodine and DHPR, dihydroxypyridine and ryanodine. That receptors regulate dash channel. Just now we saw the opposite version of the question. Option 1 sodium, option 2 potassium, option 3 chloride, option 4 calcium. What is the correct answer? Calcium only, right? I don't have to wait for this because we discussed so many times. Yeah, that's about today. Now there is only 2 more weeks left, and there will be one extra session where I will be discussing um, some more important, um, like everything, including question from all the major introduction part, second part, third part, fourth part, everything. Some questions will be again solving, and also please try to solve assignment questions. Exam is nearing up, so all the best. Um, is there any queries? Please join the sessions and we can sort it out. So thank you and see you next week, Friday from 6 to 8 with the session of week 12. Sorry, week 11. Discussing next week 11 lecture series. Bye bye.